All right. So our next speaker is someone who I've had the pleasure of working with my time at UNC, Zhang. Started out getting his BS in chemistry at Fudan University in China. From there, moved to the University of Michigan, where he pursued his PhD in chemistry and biophysics. After that, he was a Life Sciences Research Foundation postdoctoral fellow at UCLA for about 4 years. After which he joined University of North Carolina at Chapel Hill Department of Biochemistry and Biophysics in December of 2011, and since then has won or has earned an NSF Career Award and is diving deep into invisible regulatory states in RNA. Take it away, Chi.
Okay. Thanks, Brad, for the kind introduction, and thank the organizers for the invitation to attend this North Jersey ACES NMR topical group. It's the first time I'm here, and I'm really grateful to be able to share some of our research. So can anyone hear me good? I guess.
Yes, we can hear you fine.
Great. Okay. Thanks. Yeah. So first, just for disclosure, so of a conflict of interest. So I'm an advisor to and hold equity in a new startup company called Ensembles Therapeutics in Boston. So my research is working towards understand, as seen in the title, the invisible regulatory states in RNA, and I would say in the last year, more than a year and a half, I think the globe has witnessed that the bad side of RNA, all right, the COVID virus has really caused devastated global health pandemic, and also have seen the good side of RNA, where RNA vaccines have been developed in a record speed, and that has been put in real world and help millions and millions of people from dying from the virus. So in general, so this is the central dogma in molecular biology. So we know a lot about, in terms of DNA, there are genetic materials, and we have heard a lot of great talks in the morning, and there's talk about the proteins being the function machineries, and what about RNA? So besides the viruses, so actually in our human genome, so we actually encode tremendous amount of RNA. So within the entire genome, which let's say 100% of our DNA, so roughly 75 and more than 75 to 80% of our genome actually are coding for RNAs. And within this 75%, 2% of them are used as a protein coding, which is part of the messaging RNA that's been translated to proteins. So the rest of the RNA coding genes, actually they code for a variety of RNA molecules. And their functions can go from protein synthesis to scaffolding to signaling, let's say the signal recognition particle RNA in trafficking, and to the viral RNA immunity. For example, the CRISPR has been powerful genetic tools in gene editing, and the majority of RNA actually is involved in gene regulation, and also not to mention about the maintaining the DNA, the chromatin integrity of the telomerase, and also splicing, and also the SARS-CoV-2 RNA also is now widely known. So, and as a therapeutic point of view, also if we look at the human genome, so the majority of pharmaceutical industry are actually focused on targeting proteins, which among this 2% of our genome, so about 0.2% of the proteins are actually our genome are actually related to diseases, and a quarter of that actually has been actively pursued as a drug target. So there are just a lot of unknowns and a lot of opportunities in working towards RNA and using RNA not only to understand their function in biology and human disease, but also in terms of the opportunities of using them as a drug target. So if we look at RNA, so we know a lot about proteins, but RNA actually, they are really, we call them chemically simple. They so for example, they are just a polysugar in the backbone, and they have different side chains, but the side chain is far less compared to the protein. proteins, there are 20 different side chains, but in nucleic acids, there are only 4. In RNA, it's A, U, G, C. And if you look at them, they follow quite simple pattern. For example, you will have the G and the C will be base paired in a way called Watson-Crick base pairs, and A and U can be base paired. And most of the time, if you see in the literature, you will see this type of diagram where individual ladder is actually corresponding to one base pair. And of course, this is A-form helices in two-dimensional space, but in reality, they are all three-dimensional, so they fold into a 3D structures, and this is typical how an RNA look like in three-dimensional space in terms of the A-form helix. So of course, RNA you can think about if you have a really long RNA strand, and they can actually follow the basic rule of Watson-Crick base pairs and also other mismatch base pairs, they can fold into this quite a long range interactions. And encode with them, if you see, those ones are simple A-form helices, but then they have all the other different motifs. For example, you can have 3 nucleotide bulge, for example, look at here, and they'll have things called apical loop on the top, and they can have even more complex, let's say, 3-way junction and even pseudoknot interactions. And although it's highlighted here as a secondary structure in a 2-dimensional space, but in reality, you can imagine they can fold into very complex structures in 3-dimensional space. So this is actually, despite they are chemically simple, but structurally they can be actually quite diverse. And on top of that, more challenging residing RNA is those structures, they are not really static. Actually, during their functional processes, so they will actually fold into different states. So as you can see here, so in a way in RNA recognition, so the 16S ribosomal RNA, in the absence of protein, you'll have quite extended conformation. First, when it's bind to its cognate protein, it will reorganize the arms such that it will bring 2 helices closer, providing a platform for the sequential binding. And in terms of in the viral replication, here you can see the structure change is even more dramatic than just rearranging the helices. So for example, you can have a really linearized RNA, this is from HIV, and it can branch out, literally undergoing a secondary structure transition. Almost in the protein world, you can think about an alpha helix becomes a beta sheet. And this branched out structure will undergo a self-dimerization process, form a kissing dimer, and finally form into a more stable duplex, and this will be packed into the virus. So you can imagine in this whole process, RNA, not only they can form complex structures, but also they take advantage of structure changes. So if we want to really understand how RNA function, so in a way, we can't really focus on one single static structure. In a way, we have to really look at its conformational landscape. So you can think about most likely the biological active conformation adopts the lower spring energy state. But if you look at its own energy landscape, so actually RNA can literally go alternative conformations, which has slightly higher energy. And those higher energy leads to lower probability, lower population, and sometimes depends on the barrier, it can lead to an even shorter lifetime. And in the NMR world, we all know, we call them the excited conformational states relative to the ground states. And while those are maybe sounds like a conceptual concepts, but in reality, actually biology are taking advantage of this very hierarchical conformation landscape of RNA. For example, you can have positive regulators come recognize biological active conformation and stabilize further in terms to enhance its function. And you can have negative regulator actually recognize an alternative conformation and trap it down with a very deep connected trap such that you will never be able to evade the barrier, go to its a positive biological active conformation. And a lot of bacteria, actually, they are using metabolites to recognize different conformations along the energy landscape such that it can regulate its function. And also that gives us ideas about how to use this landscape to develop RNA-targeted small molecule therapeutics. So in a short summary, so in my lab, we are actually we aim to visualize and also target RNA functions. So I'm highlighting here 4 different pillars of what we do in the lab. So we are MR spectroscopists, so we develop MR techniques dedicated to visualize RNA dynamics. And also recently we have been starting incorporating cryo-EM, X-ray modeling, as well as high throughput sequencing information towards our understanding of the RNA conformational landscape. In terms of biology, where as I mentioned, so a lot of RNA function in biology are involving gene regulation, and this is actually one of the main areas we are interested in. So we are studying gene regulation bacteria as well as gene regulation in mammalian, and ultimately we would like to take our knowledge from the studies we've done in the gene regulation. Hopefully, one day we can develop some small molecule therapeutics to manipulate those gene regulation processes. So today I'm going to share with you some our recent work we published early this year in terms of gene regulation in mammalians, and how RNA dynamics and how AMR help us to unveiling those invisible regulatory states in mammalian gene regulations. So the system we are interested is in terms of the RNA systems is MicroRNA. And MicroRNA are actually evolutionally conserves more non-coding RNA, so they are pretty short. They are about 19 to 25 nucleotide length. And once they are being made, so they'll be incorporated into a protein called argonaute, form a RISC complex. And there are more than 2,500 microRNA has been identified in humans. And more than 60% of mammalian coding genes are actually regulated by microRNA at post-transcription level in 2 different mechanisms. One is this RISC complex will come and will inhibit the M7G dependent translation, or you promote poly-A degradation. Or alternatively, if the microRNA is perfectly impaired with the messenger RNA, the argonaute protein will come will execute its endonuclease activity in a way you will cleave the mRNA, and the mRNA will be subject to degradation. So while the RNA is relatively small, it's like 19 to 25 nucleotide length, but it's in order to make it, actually it has to go through a quite laborious process. So for example, the mRNA gene can be either encoded by itself or be part of other coding genes to be transcribed by RNA polymerase II. And form this really long, we call them primary microRNA. It features the M7G and poly-A tail. And in the nucleus, this primary microRNA will be recognized by a single set of microprocessing complex called DROSHA, in complex with DGCR8. And this complex, protein complex, will come and recognize this helping loop architecture of this within this thousands long of primary microRNA, and just cleave it out, become a simple 60 to 70 nucleotide long precursor microRNA. And precursor microRNA will be exported out by exporting 5 into the cytoplasm. And then in the cytosol, so dicer together with a TRBP protein, it will come and recognize the precursor microRNA and start processing in terms of cleaving the apical loop. So now you have 2 strands of double-stranded RNA, and one of them will be selected based on simple thermodynamics into the microRNA induced silencing complex RISC, and this will start carrying out the function. So one thing quite amazing is, despite there are 2,500 microRNAs, the majority of them following the same path. In a way, all of them are transcribed by POL2, processed by DROSHA and the dicer, exported by exporting 5. So not only they're becoming interesting in biology, but also they become novel targets for developing therapeutics. So you can imagine those microRNAs, they regulate more than 60% of protein genes in a post-transcription level. So they must be highly regulated. And indeed, if you have a 3-fold change in the final microRNA level, you can lead to disease, such as cancer. So over the years, the way of how microRNA being the biogenesis being regulated has been actually thoroughly studied. For example, it can be regulated at the transcription level via a lot of transcription factors. It can be regulated at the second step, the DROSA processing step. For example, the protein, DROSA and the DGCR can be phosphorylated or acetylated. And also there are a lot of DGCR and DROSA binding proteins can come and regulate this enzyme. And also the primary microRNA itself can become a template for different RNA binding proteins in terms of manipulating its interaction between the substrate and the enzyme. And in the second enzymatic step, in the dicer processing step, also a lot of pathways has been identified. For example, the TRBP can be phosphorylated. The microRNA itself, the precursor microRNA, has a lot of RNA binding proteins. And on top of that, in addition to proteins, so the RNA can be also modified by, for example, a BCDIN3D protein. This one will come and induce methylation on the RNA, as well as ADA1 mediate adenine to inosine editing, which means you'll come and modify one of the adenines and convert to inosines such that RNA becomes a different substrate. And in the cytosol, so there are endo and exonuclease activities can come and degrade the pre-microRNA to enhance or to enhance its regulation. So if we look at this picture, so actually it's all protein centric. And I have been trained as a MR spectrometer, but also as a nucleic acid MR spectroscopist. So we always ask the question is, how about RNA? If you look, so the RNA molecule, they are actually not just a small molecule, right? They are actually, it is a biomolecule. They are actually can approach the size as big as proteins. So we ask the question, as the substrate, could RNA play a role in regulation? Particularly, let's say all those highly flexible and has a alternative structures rather than those standard A-form helices, will these areas play a role? And indeed, back in 2016, for instance, Major's group, they have used computational simulation as well as mutations. They show actually by perturbing structures on this RNA substrate can indeed lead to different maturation. And more recently, for Alan's group from ETH, they have shown actually the plasticity on the precursor area actually do involve in manipulating microRNA biogenesis in plants. So we decided to take to study one of them, and the one we are particularly interested is an oncogenic microRNA. It's called MIR21. So you can see MIR21 is actually the first discovered mammalian microRNA, or the microRNA has been there, for example, in plants, in other organisms. But MIR21 is the first microRNA discovered in mammalian by Brian Cullen's lab at Duke. So this is how the primary microRNA looks like, and this is where the mature MIR21 sequence. And of course, complementary to that is we call the MIR star, is the one that's not being selected into the risk complex. So the mature microRNA MIR21, when it's being produced, so its function is mainly as an oncogenic microRNA that inhibits PTEN and PCD4 by degrading their mRNA. Also recently has been shown that MIR21 plays a critical role in heart development. So one thing interesting about MIR21, the reason why we're really interested is, so it follows the canonical biogenesis pathway. For example, it's involved in transcribed by POL2 and processed by DROSA and the dicer, and then incorporated into Argonaut. And also, it is regulated at post-transcription level. That means something happened after it's been transcribed into this form, actually plays a role into regulation. So studies by Collins and co-workers and other labs show actually if you start manipulating the pre-elementary region, actually you can really affect the DROSA processing as well as dicer. So now we want to really understand how does this conformational plasticity within this help. And of course, you can imagine we are not, probably not the first lab thinking about this. And indeed, Kevin Lupke's lab wanted to solve the structure of this, but however, due to the conformational flexibility, so they actually had to make a mutation to stabilize the pre-element region in order to solve a three-dimensional structure of this pre-element region in miR-21. And more recently, Gabriel Vrani's group, they have shown actually this region is actually quite conformational flexible and can be stabilized by a peptide. So we decide to take a look at this region. And the first thing we do is, of course, we make a sample, right? So my student made, we call the pre-E miR-21, which we take the part of the stem region and also on the top of the pre-element region. So one thing I want to point out to you, so this RNA is relatively small, so we think it should be pretty straightforward. There are only 29 nucleotides. And when we take a C13 HSQC spec, so those are the one we're looking at the base. So in a way, so here we're looking at either C6H6 from U and C's or C8H8 from A and G's. So this is almost like the NH-HSQC spec in the protein. So every single peak corresponding to one nucleotide here. So one thing we look at this, we were quite excited because it looks really clean, very beautiful spec. However, when we start counting the peaks, we can only count 20 peaks. We're missing 9 nucleotides. And that is a big problem, right? So either the RNA is degraded or something else is interesting happen. So indeed, when we heat up the temperature from 25 degrees to 35 degrees, so a lot of peaks start showing up. And indeed, we can rescue almost all the peaks. Here you can see it's you can count 28, but actually there's one more extra here. So we can rescue literally all 29 peaks. So or MR spectroscopy, once you look at this, you probably already ask yourself, oh, that means this is undergo intermediate exchange, and now we're pushing it towards a little bit faster exchange, such that we can probably study it. So one thing is, if we count them, there's a single set of resonance. That means we are not under the slow exchange where you have two sets of peaks. So there's only one set, means either it's a population average or other mechanisms. So we decide to look at it using relaxation dispersion techniques. And so the way the technique we want to use is called low spin lock field R1-rel relaxation dispersion technique. So conventionally, the R1-rel dispersion, we know it's used high power, high spin lock power, and it's very powerful in terms of characterizing fast exchange process that is above 2,000 per second rate of change. So low spin lock field R1-rel dispersion actually is really powerful. It can actually let you not only get higher spin lock process, but also study a little bit slower exchange process. And the low spin lock field R1-rel was initially introduced by our Palmer co-workers. They show actually you can go the spin lock power as low as 150 hertz. And Lewis K and co-workers, they have introduced a 1D selective R1-rel. They show actually you can go spin lock power as low as 25 hertz. And Hashiman co-workers, they have shown actually they adapt the scheme developed by Lewis K and co-workers, and they applied it to nucleic acids. And they have shown actually in nucleic acids you can go as low as 90 hertz. That has something to do with the coupling in nucleic acid. So in terms of how slow a process you can go, so K and co-workers, they show actually they can go about 280 per second rate of change in proteins. And a few years ago, we have shown actually we can go as low as 60 per second rate of change for nucleic acids by the low spin lock power, low spin lock field R1-rel experiments. So now we think, okay, so that could be a really cool technique since we don't know exactly what the rate of change, but this might provide a versatile tool for us to characterize it. So now we start using the low spin lock 1D selective R1-rel. So we characterize the system at 35 degrees, in which we can see all the resonances. So for example, we first look at the bottom helix. For example, let's say this A. So on the left, I'm showing you is the unresonance R1-rel. For example, this is from 3,000 hertz spin lock all the way down to 100 hertz spin lock. And you can see there's a flat line that indicate probably there's no exchange process. We also categorize the system using off-resonance dispersion. For example, the red, orange, and blue are 3 different spin lock powers, and we apply them at different off-resonance sites. And this is being converted, this R1 row has been converted into R2 plus REX profile, such that we can compare it to the unrest. And you can see there's a fat line indicates there's no dispersion or no conformation exchange, at least within the detection limit of the R1 row RD experiment. So we look at the rest of the helix. So all of them, they show this very flat line indicating there's no conformation exchange in the lower helix. However, once we start moving away from the lower helix, let's say we start looking at this adenine, clearly we can see a very nice dispersion profile. This is on resonance. There's a power dependence. And also, if you look at the off-resonance dispersion experiments, also we start seeing this peak showing up. And for example, the lowest spin lock power has the highest peak, and the highest spin lock will have the lowest. And also, one thing interesting is for the off-resonance dispersion experiments, you can actually tell where the chemical shift is of the excited state. For example, here it is downfield shifted. So this is just one adenine. So how about the rest of the helix? And indeed, we see a global, for all those ones we have looked at, they have isolated peaks. For example, those ones, they are overlapped, so we couldn't really measure the data. But for all of those we can measure, they all show this type of relaxation dispersion profile, indicating they are undergoing some conformational exchange. And the nice thing is, all of them can actually be fitted into a single 2-state process. In a way, there's a ground state conformation adopts 85% of the population. There's the excited state that adopts about 15% population. The lower helix, they are well formed, there's no dispersion. And the rate of change, KEX, is about 1400 per second. So now the question is, what is this? So one thing, as I mentioned, the chemical shift of this excited state, right, from the off-resonance dispersion experiment can actually give us some clue of what the alternative conformation, or we call the excited state, could be. So for example, the one from the adenine, the A22, the C8 from this residue, displayed the highest chemical shift difference between the ground state and excited state, as you can see here. There's so the excited state is about 1.5 ppm downfield shifted. And if we go through the literature, so for example, Victoria D'Souza and co-workers, they've shown almost several years ago, the marine leukemia virus actually has a protonated state of an adenine that leads to a stabilized pseudonode structure. And that one has a downfield shifted C8. And also recently, Hashim and co-workers, they also have observed a protonated adenine that has a downfield shifted C8. So we think, okay, so that might be the case. So in order to prove that or show some support of it, so we decide to change the pH from what we normally use, pH 6.45, to a higher pH, pH 8.04. And indeed, so this A22 peak starts showing up. From the severe exchange broadening, actually it becomes very well observed. So now with that, so we can also now start asking, so since we can see this A22, the C2H2 residue, how about if we measure dispersion profile on this? And what we're able to observe is a massive chemical shift change. It's an upfield shifted by 8 ppm. So that indicates one of this neighbor is protonated, such that this carbon can have a very high upfield shift, chemical shift. So the question is, which one is it, or is it due to the protonation event? So in that sense, so we like to switch gear to a CEST experiment, because CEST experiment, like R1 row, it can be used for go to very low spin lock power, and actually can go even lower than R1 row. And it's been very powerful in studying slow exchange. And the CEST experiment, it's originally, it's back in the 60s, being developed by Forrest and Hoffman co-workers. And in recent years, Maris Krohn co-workers, they introduced the concept of CEST in terms of testing, studying in fibers, and loose skin coworkers, they have bring CEST into a solution protein spectroscopy, MR spectroscopy studying excited states. And we recently also has been working introducing CEST into nucleic acids in terms of carbons. So now if we want to look at the nitrogen, so we also a few years ago, so we actually first we take the similar approach as done by Kay and coworkers. That's just for a regular NH protonated nitrogen. We can monitor that. And also in this particular case, we are interested in looking at those nitrogens they are not protonated, and that is the one we are particularly interested in look. And in order to do that, so for the non-protonated nitrogen, we have to introduce a way that's slightly different from the protonated nitrogen, because the J coupling is relatively small. So in this case, we are not really preparing a pure NZ magnetization. Instead, we're preparing a two-spin order NZ-HZ magnetization. And this is a similar approach that has been worked by Kay and coworkers, and also Pei Zhou and coworkers. They have used for the proton that link to the amine nitrogens. So in this case, we also take advantage of a different spin label approach. For example, as you can see here, if we are having a double labeled nucleotide, for example, if you have all nitrogen and carbon labeled, we actually have quite low signal noise. Versus we only label nitrogen alone, we can have tremendous high signal from the 2J bond coupling infecting CEST. And this phenomena is also reminiscent of Kwaku and co-workers. They have introduced to look at the carbon CEST, in a way have a site-specific label carbon to enhance its signal noise. So in this way, we think having a nitrogen only labeled, because now we don't need to worry about the carbon cause a signal loss to its neighboring nitrogens. So this really provides us approach to get a very high sensitivity. So now we apply this approach, right, this non-protonated nitrogen CEST to study the MIR-21. And one thing, as you can see, so we only observe an excited state of the nitrogen from the 822 of N1. And this looks like a typical CEST profile. But I would like to ask you to pay attention to the chemical shift differences. So this is almost 70 ppm upfield shifted. And that is consistent with you have nitrogen that's being protonated. So it's shifted from non-proton 220 ppm all the way to a protonated nitrogen that is close to 160 ppm. So together, so now we are able to combine the carbon R1 row as well as the nitrogen CEST to confirm this is the only site that is protonated, and it's protonated at N1 location. And since as MR spectroscopy, we all love nitrogen. Oh, sorry, we all love proton. And this proton, additional proton, is actually it's the smallest chemical modification you can think about in any biomolecules. And just to confirm those two different techniques, R1 row and also CEST, they're comparable to each other. So if you can look the excited state population from R1 row and CEST, they are really comparable to each other, almost identical. And also the lifetime of the excited states, they are similar to each other within the error. So now we can actually also use this to categorize what is the pKa of this this adenine and why it's shifting. So for example, we can measure as a function of pH. And here we're monitoring the R1 row dispersion profile of this adenine. As you can see, the lower pH we have, the higher the R1 row, the R2 REX peak is. That indicates it has even higher population. And indeed, we can fit all of them, and we can see there is a major pKa shift from a 4.17 for an adenine that is by itself, all the way to close to pKa of 5.8 for this structure RNA. So now we ask the question is, okay, so there is a protonation event happening at this RNA. So is this the driving force for this? So number one we did is we delete this adenine. So now there's no protonation site. What are we going to preserve? So indeed, when we measure the R1 row relaxation dispersion both on and off resonance, we observe no exchange in the entire system. So the second we ask is, how about this really weak stem, which is GU and UG base pair? So we stabilize it by a GC and CG, we just mutate the U to a C, and also we observe there's no dispersion at all. So have a stabilized helix also kills the exchange process. And the last one we think is, how about this really big apical loop? Could this also play a role? So we mutate that big apical loop to a well-known stabilized tetraloop called UUCG tetraloop. It's a C-UUCG tetraloop. And as you can imagine, so now it also suppresses the exchange throughout the entire system. So put those all together, so you can think we can derive a model where this entire pre-element region, starting from the bunch to the stem to the apical loop, so they are actually one entity. So they undergo a concerted motion rather than a single residue plays a local motion. It's the entire thing undergo a reshuffling process. So now the question now relies, what is it? How does it go from here to an alternative state? So if we look at that, so in order to do this, so we take a strategy that's introduced by Hashimi and co-workers back in 2012. They called it Mutate and Chemical Shift Fingerprint Strategy, MCSF. The idea here is you take the RNA sequence and you put it into a program called MCSim, which is developed by, for instance, one major group, and you will predict all different alternative secondary structures. For example, this is predicted from the most well-known program called Enfold, predicting the secondary structure. Here is you are using the MC-fold to predict, and you can see they are quite different between the ground state we are hypothesizing. And one thing quite interesting is you can see all these G's are actually base paired with this A22 as we can think. So now, if that's the one we want to focus on, the MCSF, the mutation and chemical shift mapping and fingerprint strategy gives us a protocol where if we can stabilize this excited state conformation by mutations, for example, by making this G to a U, so now this is AU base pair, so you can stabilize it, and now we can compare the chemical shifts of the excited state we obtained from the ground state to this alternative state. And indeed, for example, I'm highlighting 3 different nucleotides. One is from the A22, it's C1 prime, which is from the sugar, also a G28 from the sugar, and also a 30 from its base. So for example, you can see here, this is the ground state, this is the measured. So this is the ground state, and this is excited state. And we compare this excited chemical shift to either a mutant, which is G38, we trap it down into this excited state conformation. It's in a good agreement, as well as we can actually lower down the pH if we think this is the state. So we can lower down to the pH, and also you can see the excited state is in good agreement with this lower pH chemical shifts. So this tells us, so maybe the excited states adopt a non-canonical A+, a proton A, anti, and a G-syn base pair conformation, like what's shown here. However, we would like to have even more evidence to support our proposed model. So in this case, we can see the ground state has 85% population, excited state is 15%, right? So that's why it goes here about 120 per second, and it goes back much faster. So we'd like to reverse this population by making this one as a ground state. So in a way, we can actually change its pH. Since it's protonation dependent, we can change its pH to 4.35, such that this excited state at the neutral pH becomes the ground state, the most popular state. And we can then apply the conventional NMR as in a nosy spec to confirm whether this A plus G forms the proposed A, the anti-A and the syn G conformation. So indeed, if you look at this nosy, we have a cross peak between the bases. For example, we can have this H2 from the adenine A22. So for example, those are the HSQC spec. So we can identify exactly where the C2H2 of the adenine 22 is. So you can clearly see there is a strong cross peak between A22H2 and G38H8. And this can only be possible if the G adopts a syn conformation where it flips. So you can bring the H2 and H8 very close. Otherwise, in the canonical anti-anti conformation, the H8 will be too far away to show any cross peak to the H2. And to further confirm this, we can also look through the backbone. For example, the proton of the previous base to its own to the next H8. If it's in the syn conformation, you won't be able to observe that. Instead, you observe the G28, it's H8 to the next, then +1 H1 prime, the sugar. Versus in a canonical anti-conformation, what you'll see is you will have the base proton H8 to its previous H1 prime, the sugar. So indeed, if you look at the spec, so this is the cross peak we're able to observe. And indeed, if you look, this is what we would anticipate if the G38 adopts the canonical anti-conformation, but we don't see any peak here. Instead, we see a very nice cross peak between the H8 to its next H1 prime, which confirms a syn conformation for the guanine. So we have talked about so much about anti and syn. So what's so unique about this A plus anti G syn conformation? What's unique about this mismatch? So now if we look, this is how if we want to base pair them together, and this is where its sugar is. So quite interesting. So if you look at its sugar, we use, in a way, if we think about A-form helix, it's a backbone to backbone distance. If we use a C1 prime as its metric, as its index, so in the anti-A and the syn-G conformation, you'll have a C1 prime distance about 10.4 angstrom, which is a cross helix. You can think about in the diameter of the helix. And this is actually identical if this, instead of it's a plus G conformation, it's a regular Wilson-Crick anti-A, anti-U conformation. They also maintain a interhelical distance about 10.4 angstrom. However, if we think about if A and G does not form this anti-syn, instead of it's anti-anti, so the interhelical distance is actually 12.9 angstrom, which means the helix has been widened quite dramatically. So in a way, so this anti-syn conformation by protonating A, it maintains its A-form-like geometry for the entire helix. So what's unique about this, or what could the functional outcome be for such a protonated induced A-form geometry? So we have talked about regulation of the dicer process where you can manipulate the proteins. Actually, indeed, in order for dicer to recognize and to process a substrate, there are certain optimal geometries or certain optimal topologies for those type of substrate. The first is both cleavage site. So dicer cuts the RNA, right? So both, and it cuts on both sides. So both cleavage sites, they have to reside within a double-stranded A-form helix. And second, both cleavage sites are positioned 2 nucleotides away from a flexible loop. And if we look at the excited state, actually it perfectly matches what's an optimal substrate topology required for dicer. For example, by having protonated A has an anti and G conformation, so this entire helix adopts A-form helix, which is a double-stranded A-form helix. And also by base pairing this, it's going to induce a GC base pair and also UU base pair, then followed by a big apical loop. And this large flexible loop is indeed capped by 2 base pairs away from the cleavage site. So that gives us an idea, maybe the excited states are actually a much better substrate for dicer. So in order to validate our hypotheses or to support our hypotheses, So we decided to actually carry out a dicer processing assay. So this is the Y type. So on top of the Y type, we also designed two mutants. One we called a ground state mutant, we called it Premier 21GS. In a way, we stabilize this GU and UG base pair by GC base pair. And the other we call excite state mutant, mimic mutant, where we, similar as what we have done, we make this G, we mutate this to a U, such that this AU base pair can be formed. And this will adopt a similar excite state secondary structure or tertiary structure from the mutation. So we can then carry out a single turnover processing. So this one, we are not really studying the load and unload process. We're just trying to understand how does the geometry of the substrate really helps dicer process. So in a way, we are carrying out a single round turnover. So this is at normal pH for all those assays, pH around pH 7.8. So this is a precursor microRNA. And when it's being cleaved, it will generate a mature microRNA. And also you can see here, there are bands. That means it's either cut on one side or cut on the other. So if you can see, we have carried out 3 different substrates. And clearly, just visually you can tell, the excited state substrate is much better substrate than both the Y type and the ground state mimic because it's being processed readily. And also, since the pH matters, so we also went down lower pH, pH 6.5, and also we measured the processing. And still, as you can see, the excited state mimic is a much better substrate. So since we are here doing a time dependence, so we can indeed actually fit out the processing as a function of time, and we can actually carry out to obtain the observed catalytic rate for both the ground state and excited state. So as you can see, actually at higher pH, so the ground state, the excited state mimic is actually almost 4 times better in terms of processing than the ground state. When you go to lower pH, it's actually shown a much bigger difference. The excited state mimic barely changed, but the ground state mimic has been actually highly suppressed. And the excited state mimic is actually has a more than close to 28-fold enhancement in terms of processing. And then we start working on, in a way, we can plot the ground state and excited state as different time points. And if we think the Y type is a population with average of the ground state and excited state mimic, so we can, since we have the population derived for both pH conditions, we can actually just measure, calculate a population with average by taking those two. And then we can compare that to the Y type. And indeed, the population with average showed a quite good agreement with the Y type, which indicates, so in the test tube, the Premier 21, in terms of this 4-length construct, actually undergo 2 different populations. And once one population be recognized by dicer, it will be committed to that process. So in a way, so in a short summary, so we have shown actually even for the, you can think it's a really simple RNA. Actually, it's not quite simple. So actually, it encodes an environmental sensitive dynamic ensemble in response to proton, which is acid, and it can manipulate its conformational transitions and control the maturation. And in terms of what's the biological relevance, so as most of us can recognize, so for cancer, due to its changed metabolic pathway, cancers are actually super acidic. Of course, it's a microenvironment acidic, and you can imagine during onset of cancer proliferation, so there's an insight change in terms of this metabolic pathway can promote a rapid flux in terms of generating acid, and that might be coupled with the process of generating a highly optimal substrate for dicer processing to generate miR-21, which together with all the other factors promote cancer development. And with that, I would like to summarize. So I've shown you a story I've done recently, and hopefully in the near future, we can have the opportunity to share with you in other process we have done. And finally, I would like to thank my group So the work I've told you is done by Jerry Bankston. So he recently graduated, and he's a postdoc in Scripps. And I thank our facility manager Stu Parham for maintaining an excellent MR spectroscopy center for us to use and to categorize all those functions, and our collaborators and the funding. And thank you for your attention. Oh. Hi, Christian. I think you're muted. You're muted. Sorry, you're muted.
Okay, I did not know whether I can just ask a question.
Yeah.
It seems there's no chairman at the moment.
I know.
I was wondering, I mean, these this A, which was in this little bulge, I mean, how common are such elements? I mean, this very detailed study or so could have potentially a more general relevance or so. I mean, can you now search for such elements that could do similar transformations or so based on pH and so on to maybe generate more general rules for RNAs?
Yeah. So that's a great question, and indeed, I think it's general. And actually, the proton A is not. We're not the first one to observe them. For example, Jim, they have observed them. Victoria D'Souza and co-workers, and it's quite well known in terms of even crystal structures have observed the proton A from an AG base pair. That's where we get the distance, which is from the crystal structure. But those are all isolated cases, right? So, and also sometimes they are involved in much more complex structures. For example, let's say Victoria D'Souza structure is a pseudonaut. You might think, oh, that's probably pretty cool. And Hashim's is also in a complex, the iris. So ours actually, it's really simple. We were quite surprised. Indeed, we actually now we have more systems in the lab. So we actually also observe there are all those protonated adenine. And it's in general, sometimes because, for nucleic we always work on like pH 6.5-ish. So sometimes they are just not, they could be broadened. And many times when they're broadened, people stop studying them. And then they change the pH to high pH and they look really nice back and they just forget about, there is a broadening peak. So we actually have studied in the lab several other systems where indeed actually at like say neutral pH or 6.5, we see massive broadening, and we go to higher pH, we see really nice peak. So now it's become a general protocol in our lab is at least do 2 pH, and we will monitor particularly on the C2H2 because that is the one because the chemical shift change is about close to 8 ppm. So that gives you the most sensitive probe.
Now, if I can extend my question, I mean, since you started also with saying that excited states might then be trackable. Now, if these excited states have more general structures, then you might have problems with specificity. So what are your thoughts on that?
Yeah, so if we want to drug the excited states, so one thing is, RNA, if it's just regular A-form helix, you are totally right. I mean, the selectivity is a big problem. However, we think, once, let's say you have those weird chemical modifications, right? Even it's a plus G, and together with the rest of the up group with different sequence, there might be a chance to find some unique structures. And that's one thing we're trying to do here is, so as I shown you, it's all secondary structures. So we don't know the tertiary structure, and based on the relaxation dispersion measurements, we know the excited state actually has its own kinetic it's a kinetically stable state. It's not IDP. It's not intrinsic disorder. So there's a certain set of structures. And for example, when we measure NOEs at low pH, we do see NOE cross peaks.
Okay.
So now what we're doing now is trying to solve a structure at low pH, but at low pH there are also other issues where other, let's say, cysteines, the cytosines, right, the C, are also start getting protonated, and then it's just a question of whether that structure at a low pH is a true structure or not. So what we're doing now is we combine NOE as well as chemical shifts, because recently Hashim and co-workers and also David Case, they've shown actually chemical shifts contains a lot of information, can help you define the tertiary structure together with the Rosetta-based, let's say the FAFAR, the Rosetta-based computational structure prediction programs. So we're trying to take a bit of data from different approach, because there's no clean, let's say, low pH. You get NOE, but then there are other modification, protonation occurs. High pH, you don't get the protonation, but you can use the RD to get the chemical shift information. So yeah, so now we're trying to get all of them together. And also the other thing we're working on, and a few years ago, we published the pulse sequence to measure RDCs. We love RDCs, right? So my graduate study is all about RDCs. So we are still working on RDCs. So we published the pulse sequence. Actually, that was in light of Lewis case sequence. So we can measure RDCs of the excited state. So now we're using the excited state RDC together with chemical shifts, everything, in order to get a structure.
Okay.
And I think maybe that can give us some clue. But you are totally right. It could be a very challenging.
Thank you very much.
Yeah.
Hey, Chi, question. So these like adenine protonations and anti-syn interactions, are these more common in like bulges and loop regions, or is there a world in which they also can exist in the more structured like double-stranded part of the RNA, and that could potentially lead to like alternate processing or alternate folding in pH-dependent manners?
I think so. Yep, it can happen. It's just in our case we observe in the stem, right? In the bulge, but of course when it's formed, it's in the stem. But you can think about, you can have something which is a really long helix on both ends, and you can have the AG and the G can flip and form a protonated state, because that does provide you a better stacking interaction and a less distortion on the backbones. So think about if the helix, right? If you have to get the AG in the canonical anti-anti conformation like this, the helix has to bulge out, and there will be less stack interaction. So it will make sense if it can flip, but we haven't studied that. But we do have other examples, other protonation states that within the helix it can happen, but not the A plus G. But I think it could, certainly.
Cool.
That's a great question.
Any other questions online? Feel free to unmute and ask. All right, looks like no other questions. Okay. All right, great. Thank you, Chi. Really appreciated that talk.
Yep. Thanks.